Found in virtually all human cells, this protein is essential to a wide range of dynamic processes. The role of actin can best be observed in one of the body's many mobile cells. Here, the cell uses a branched actin network to create wide protrusions, known as lamellipodia, which extend the cell's leading edge during migration. As the cell explores its environment, narrow protrusions known as filopodia, composed of unbranched actin, reach out and perform sensory functions to help direct the cell. From the essential contact between a T cell and its antigen-presenting cell, to the dynamic process of a macrophage engulfing foreign invaders, the role of actin is all-encompassing. So much so that infectious pathogens have evolved to use actin to their advantage. Infectious disease is a primary cause of death worldwide, and many infectious pathogens force entry into their host cell to evade the immune system. Chlamydia, the most common sexually transmitted infection and the leading cause of infectious blindness, uses a branched actin network to invade its host cell. Upon contact with the host, chlamydia secretes effector proteins into the cytosol. This indirectly triggers a signaling cascade, leading to the activation of the ARP2/3 complex. ARP2/3 is essential in creating branched actin networks, such as those seen in lamellipodia. Once activated by nucleation-promoting factors, this complex binds to pre-existing actin filaments to nucleate the formation of a new filament. Subunits are then added onto this growing filament, creating a branched network that pushes the membrane forward. A complex that results in movement in a normal cell has now been hijacked to allow the pathogen to invade the host. Once in the cell, other pathogens manipulate actin to propel themselves around their host and facilitate spread. Rickettsia is an intracellular bacterium responsible for a number of serious diseases, including Rocky Mountain spotted fever. It is transmitted to humans via arthropods, and it uses unbranched actin for motility. Recall that filopodia are also formed by unbranched actin. These unbranched filaments are created by formin. Membrane-bound formin proteins polymerize actin to create an unbranched network, using actin-binding profilin proteins to construct parallel networks of filaments. Rickettsia expresses the protein SCA2, which mimics the function of formin. A protein normally used to create structures like filopodia is now imitated to produce an unbranched actin tail that propels the pathogen forward. Other pathogens travel through the host and spread to other cells by further exploiting ARP2/3. Listeria, a foodborne pathogen that is most likely to infect pregnant women, older adults, and those with weakened immune systems, uses branched actin for motility. Listeria expresses Act-A, a protein which mimics nucleation-promoting factors. Recall that these factors are normally required to activate ARP2/3 to create a branched network like those found in lamellipodia. Imitating these factors allows Listeria to create and use this branching network for motility to infect neighboring cells. Understanding how pathogens exploit actin has helped to elucidate numerous cellular processes, including those involved in cell motility. In addition to its role in pathogenic disease, actin plays a key role in a multitude of other diseases, ranging from cancer metastasis to Alzheimer's. Further exploration of the host-pathogen relationship holds promise to shed light on new therapies and will continue to illuminate the expansive function of actin.